Enzymes are some of the coolest parts, proteins. They help the body's chemical reaction. They can break the bonds between the molecules. They're unique for each reaction and give a great control of what goes on in the body. We talked about that. Enzymes are named by their functions, such as lipase, which breaks down lipids in the gut. What's the suffix used for enzyme? Anybody? ACE. That's right. So if you see the word ASE, you know it's an enzyme. It's a biological catalyst, which means if we have a substrate, like this is a maltose, so this is glucose and glucose, we have an enzyme probably called maltase. I would assume that's probably wrong, but that'd be a good name for it. That molecule goes into that, the shape fits, it's shape dependent. That's why when a protein becomes three-dimensional, it's about the shape. And in this shape, there's a part here that the enzyme can eat away on that bond that's in here between. And the product will be two glucose molecules that bond. The enzyme is not destroyed in the process. The enzyme can be reused for more things. Same process, always the same process. Since this is about shape, it's very specific. You cannot have this enzyme do anything else other than break it down to the maltose. When we graph that quality, we see here the enzyme is looked at, it puts energy into the, well, when we want to like break the albumin in the egg, right? We have to put heat in it. We have to increase the heat until the bond gets broken. So that's an activation energy, we call that. And in a graph, an enzyme, it lowers the activation energy. That's what they talk about. So the reaction might take place on its own. But with the help of the enzyme, it's not as much work to get it more going. Which, of course, makes the fact that it happens. So that's where enzymes are very good. If you have some enzymes, like in the stomach stuff, you have enzymes that destroy protein. Pepsin. You can't have always active pepsin around if you have the gut line that has protein on it. It's going to destroy your whole protein. So some enzymes are very, they're very powerful. So you make them as an inactive form, like in the stomach, it's pepsinogen. And then you see the GEN suffix, it comes from Genesis, creation. And so that's an inactive form. So pepsinogen, for example, is inactive, and the HCl, so we add on the HCl, makes it, activates it, and then we have pepsin, which is one of the enzymes that doesn't have the ASE at the end, so just so I think that's the example. That's just to that. Good? Okay, so we have these two suffixes, ASE and the GEN. Yeah, let's talk about that. How about or whatever? How about Harper? Proton inhibitor type stuff. That doesn't let the stomach acid get made, so it doesn't go into your mouth and it burns you like this Harper. So you vaccinate. So you take a medicine that doesn't make HCl, which is a lot of them. And I forget the name, it's purple pill and all that kind of weird stuff, right?
So do you know this guy, Professor? He's a verbally like a homeopathic doctor. And my husband was on those blockers for 2-3 years. And I went through a few, they're putting me in, they need like a lifetime supply, and I had no way. And his theory is very different about why people have heartburn and stuff. That is used to the stomach. It's off-gassing, and that's what blows the valve open and allows the stuff to come out. Because stomach acid is so incredibly necessary for the stomach.
Oh yeah.
And your stomach's supposed to be sterile. Nothing's supposed to be growing in there. That's another thing stomach acid's supposed to do.
Kill it.
Kill
Kill everything. Yeah. But think about it if you don't have it. Think about if you take one of these things, one of these pills, that doesn't make that HCl be made. Yeah, number one, you don't make pepsin. It keeps pepsinogen up there. Pepsinogen is not reactive, so the protein is not really digested.
And they say, right, people that are on those pills don't get a lot of nutrients from their food.
I know.
Right, that's it.
And the stomach is basically after eating the mouth or some, you chew it up and you have some anyways in the stomach, it's really happy. In Chinese medicine, that's the first thing to go. To look at in terms of normalizing and making better. And so we have to be very, very careful. I know the symptoms horrible, but like a pepto liquid that pushes the acid down away from the esophagus, or a Mylac or something, that seems to be much more sensible because you don't not make the HCl, you just sort of wash it down when it's too heavy. Because it isn't, of course, it's not anything you want to have. So that just brought that one up real quick, I'm sorry. But it was a mystery to me for a long time, so I thought I want to share it since I got enlightened on it. Alright, let's go to this DNA thing. You heard of that thing probably. It's part of the cell. Holds information on how to make different types of protein. So it's a protein cookbook. Make different types of protein. In fundamental makeup, it's a strand of 4 different molecules that repeat themselves. They are arranged in specific sequence, and the sequence of those molecules will code for the sequence of amino acids that makes up the protein. Whoa! You got that? Not yet. Okay. Well, let's talk about first. What are the 4 nucleotides we have in DNA? Anybody? Good. Adenine, guanine, cytosine, and thymine. So that's all we got in there. Now, how we arrange them is going to be determining what kind of protein we're going to make. So we're going to arrange them. We say, well, we have an A, we have a T, we have a G, we have a C, we have a T, and then yada, 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 and we take that sequence, the whole thing, and we know that makes a protein. It's going to be a G. Yeah. 3 of them in sequence will be translatable for the body into an amino acid. So that's very, very useful for us. Okay, when we look down here, we have a double helix. So that's 2 strands. One on one, well, then they rotate. So we have 2 strands that go up on each other, and they are having the nucleotides facing each other as the rung of a ladder. And we have specific nucleotides that can face each other. And the way that they face each other is that adenine will always touch a thymine. The guanine will always touch a cytosine. So they always work together in those pairs. They call them complementary base pairs. And we have to remember that. So I think A and T go together, and C and G go together. Well, C and G is both rounded letters, so that helps me with that. But A and T is like H and T. I remember that. And I don't like H and T.
What's uracil?
Uracil is good. When we make a protein, we have to take the part of the DNA that codes for that protein, that has this sequencing for that protein. We have to take that from the double helix. First, we have to unzip it to make sure we have 1 strand, and then we copy the strand. Because this is very sacred. We got 2 make helix. We don't want to destroy that sucker, so we want to make a copy. And then the copy goes out of the nucleus and makes a protein. And so the copy is called an RNA. The copy of the DNA that will make a protein. And in an RNA molecule, one of these complementary bases gets exchanged. And in an RNA, the thymine becomes a uracil. And what?
I was thinking RNA is the only one that has uracil. DNA doesn't.
Right, DNA has not. DNA has thymine, RNA has uracil. So in a test, for example, if you're asked a question, you have a sequence of nucleotides, and you ask which of these is an RNA and which is a DNA. And you have one that somewhere in there has a U.
RNA.
You know it's RNA. There you go. Got one right. It's on the test. Oh, this is also a computer. But does that fundamental concept so far make sense with that DNA? Because we're going to take it on and we're going to make protein out of it. So we have a few more processes that come with it. Okay? But the fact that the nucleotides in sequence code for amino acids that then make protein, that's a fundamental law. Because that brings me to ATP. That's adenosine triphosphate. It is the body's energy molecule, which is very important for us. What we need to know about that molecule is the three phosphates that are attached. Adenosine triphosphate. So that's important. Breaking the bond that attaches the third phosphate to the molecule liberates a great amount of energy that the body can use for work, which is muscle contraction, for example. Once that bond is broken and the energy is released, the whole molecule only has two phosphates. What do we call that molecule? Huh? Anybody? ADP. Adenosine diphosphate. And that's a very ubiquitous molecule in the body because guess what? We use a lot of energy. So when you look at the energy in the cell, we take this ATP. So see, we don't care about the A, we just call it an A for adenosine. We don't worry about the chemical formula. But what we worry about for this class is this release of energy when we break that bond, that third bond. We break that here, that's the energy. From ATP to ADP. And then when we eat glucose and it goes through the system, breaking down the glucose, goes into the cell, goes into a place called the mitochondria, which is a place where we make ATP. The food then, the glucose there, gives us the energy to reattach that third phosphate. That's what happens. That's what happens in the mitochondria. The process goes back and forth. We also have a molecule called AMP. Guess what that stands for? There you go, adenosine monophosphate.
So does all of this happen inside the cell?
Yeah.
The phosphate breaks off,
All that.
but it's still around.
The phosphate's still around.
The one
So we
the
can.
one that's broken off is still inside the cell.
So, like for example, you've got a muscle contraction. You've got protein threads in the muscle. And the muscle contraction, in essence, is very simple. You've got two protein threads, one lays there, the other one grabs it and pulls on it. And that way the muscle shortens. So whenever you have that thing attached, well actually the head, that one molecule attaches, you pull, you move that section, that joint, and then it detaches. When it wants to be brought back to the active form, it has to liberate an ATP. It's all inside the cell. The phosphate is still right there.
So you're saying to recover from a contraction, you've got to get that phosphate
Back up. Yeah. And so that all happens right around in the area. You know, and then we have
Very local.
Very local. Yeah, I mean the most ATP we can create in a specific organelle called, which is a cellular organ called the mitochondria. Then we make like out of one, whatever, we make a huge amount of ATP. We make 30